Morning, everybody. Welcome to day 3 of CampyUK Goes Global 2021. My name is Lisa, and I am going to be co-chairing session number 4. So this is the last of our scientific sessions for this conference before we move on to the fun of this afternoon's industry panel and, of course, our plenary panel. So it is my absolute pleasure to introduce Evangelos from the University of Bath, who will be co-chairing this survival and application session with me. So Evangelos is going to be introducing all of the speakers and leading the questions. If you have any questions for any of the speakers, do please put them into the Discord channel. If you would like to ask a question live, you can use the raise hand function on Zoom, and we will try and get around to you. We have limited time for question asking in this session, so if we don't get to you, then please do type your question on Discord, because the speakers can answer them after their presentation. We have a mix of 10-minute talks and lightning talks. And for the lightning talks, we are going to be taking a question for each speaker immediately after they finish their presentation. So do please pop them into the end. If we've got time at the end of the session, then we will come back. So I am going to hand over to Evangelos, who is going to lead the session. Thank you, Evangelos.
So hello from me as well. So welcome everyone in today's session. The first speaker is going to be us, Griff, to the next speaker. So we have Emily Stoakes. So Emily is a postdoc from at the University of Cambridge, and she's also at the Quadram Institute of Bioscience. And her main focus is on survival on Campylobacter species using transposon libraries. So welcome, Emily, and we're looking forward to your talk.
Yeah, so my main focus is normally on transposon libraries, but today I'm going to talk about a side project which I've been doing with Tapali and others in Quadram. So I'm going to talk about the integrated computational and experimental study that we've done to look at substrate utilization in a strain of Campylobacter called M1cam. So first up, this is very much a collaborative project. These two here, so George and Tapali, have been really integral to this study. So Tapali is our genome scale metabolic modeler. So I'll do my best to explain what those are. And then George is our biostatistician. So for any metabolic model questions, it will probably be Tapali answering those. So I'm going to take you through the 2 assays that we used during this study. So the first is Phenotypic Microarrays. So these are biolog plates, and they are pre-configured 96-well plates where they put an individual source, nutrient source, or a chemical in each well. So the plate that we used was PM1, and this has 95 different carbon sources and 1 well with no carbon source in. And it's a phenotypic assay. So the way it works is that you mix your bacteria with a tetrazolium dye, and the tetrazolium dye is reduced if there are bacteria respiring and reducing NADH. So it's showing that your cells are metabolically active and respiring, looking on certain carbon sources. So if you look at this plate, the ones that are turning purple are the sources where Campylobacter are respiring. So genome scale metabolic models. So this work was done by Tapali, and she previously published this paper here last year, where she used her model to define a medium for Campylobacter jejuni M1cam. So a model is a reaction network built from enzymes encoded from the genome, but also it allows you to take into account metabolic interactions of the environment as well. So in Tapali's model, you can take into account the nutrient source you're interested in, the gas mixture we're using in our cabinet, and also the media if it's defined. It also allows analysis of the entire metabolic system of the bug rather than just looking at individual pathways. So the models are really good because you can use them to identify mechanistic links between cellular phenotype and metabolic genotype. Sorry, so onto the background of the project. So it's known that Campylobacter can use hydrogen as an electron donor to fuel the respiration chain. And it's been hypothesized that Campylobacter forms part of a hydrogen sink within the chicken seeker and might be the reason why it's so prevalent there. So just a background into the strain that we're also using, it's a rarely documented case of chicken to human transmission. So we were interested in this study to look at the effect of hydrogen and non-hydrogen conditions on the utilization of substrates for Campylobacter M1cam. So to explain our experimental setup, we mixed our M1cam suspension with the tetrazolium dye. We then put our suspension into every well of the plates. And at time 0, we measured the absorbance of every well and also counted the number of viable cells through CFUs. We then took these plates and incubated them under hydrogen and non-hydrogen containing conditions. So you can see our gas compositions there. The reason they're a bit different, well, they are different, is because the one with hydrogen was done in our Don Whitley cabinet, where we can specifically control inputs of the gases. Whilst the one without hydrogen, we had to do in Campy jars because the Campy Gen packs don't make hydrogen. So after 24 hours of incubation, we then remeasured the absorbance of every well. We did CFUs for 23 predetermined wells, and we also dipped a 1 microliter loop into each well of the plate and streaked it onto a quarter of the plate and scored it for growth. So using the predetermined CFUs, we could create a Bayesian linear mix model, and using this model, we could predict the number of cells which were present in the 1 microliter loop and estimate it. So this allowed us to look at every single substrate, so all 95 substrates under both conditions. So first up, it's not surprising that M1cam can use hydrogen as an electron donor, but both our genome scale metabolic model and our assay under hydrogen conditions confirmed this. So on the left here is our plates that actually came out of the experiment. So this is the non-hydrogen containing conditions, the top, and you can see that there are many wells where there is no respiration happening. The well A1 here has no carbon source, so it's almost like a negative control. And you can see that Campylobacter can't respire, well, M1cam can't respire without any carbon source present. You can then look at our hydrogen plate, where if the plate is incubated with hydrogen, every single well turns purple, and that's because the hydrogen is in the atmosphere, so Campylobacter can respire. And looking at the A1 well here, so that's the one with no carbon source, there is respiration happening. This is also confirmed by our model, where Campylobacter jejuni M1cam can produce ATP in the absence of all other organic substrates. So it's important to note with this assay that it's not a growth assay and there is no biomass production happening. It's more like a survival assay. So you can see from our CFUs here is that there is an input, and then the CFU amounts drops a lot after 24 hours. And you can see there is a slight difference in the fact that there are more cells surviving under hydrogen conditions rather than non-hydrogen containing conditions. So how does hydrogen utilization compare to other substrates? So this is a scatter plot looking at the absorbance, so the change in the color over the 24 hours, and then also CFUs, and it's a ratio with the starting concentration. The negative control sits here. So the negative control is the non-carbon, there's no carbon source in the negative control, and hydrogen sits up here. So I can sort of see 3 groups here. So this is group 1, and these are the substrates where there was a change in color in the biolog plate. So there's an increase in absorbance over that 24 hours. And then there is also an increased number of cells surviving when you compare it to the negative control down here. The second group is this group here, so to the right of the negative control. And these are the ones that had no color change at all, but there are still an increasing number of cells surviving compared to the no carbon source control. And then the final group here is to the left of the negative control, where there was no color change and the number of cells were also, there were a decreased number of cells compared to the no carbon source. Sorry. Having issues changing my slides. There we go. So how do our results fit in with the metabolic model? So our biolog plate showed that 15 substrate within the plate could make Campylobacter jejuni M1 respire. Our model predicted 18. So we decided to go look at the CFUs to see what the differences were. So on this graph, there's your input of the number of cells. The output, all of these are the outputs. This is the no carbon source. The purple ones are the ones that agree between the model and the biolog plate. And the blue ones are the ones that were only found in the biolog plate, but not in the model. And the greens were only found in the model, but not in the biolog plate. So the non-significance relates to the no carbon source well, not the input. So the blue bars are the substrates which came up in the biolog plate, but not within the model. And the reason that this might be is because the model depends on annotation. So if we don't know that there are transporters or transporters aren't annotated, the model therefore can't predict these. Also, the model can't take into account certain things. So we can see here that succinic acid, the model knew it could respire using it, but here, when you add on the bromo, it can't. And this is because the model can't handle bromo yet. So then you look at the ones which were found in the model, but not in the biolog plate. And you can look at these. So these are citric, acetic, and proline. And these are potentially not being called up, and I don't know if this is right, but if you look at the biolog plate, you're only looking at NADH reduction. So even though these cells are clearly surviving, we might not have a color change because we're only looking at NADH reduction, and there are potentially other reductants happening. So glycolic and glyoxylic acid, this is because we think there are transporters of these, but we might be wrong. So obviously survival is not happening with these at all. And finally, there is L-serine, which I think is a really interesting story. So in the literature, L-serine, it's known that Campylobacter can respire using L-serine. And when we first started this project, it was true. So we were getting a purple color change. And that's because biolog plates, they normally ask you to put in BSA. And we took BSA out because the model can't handle it. So with BSA, we were getting a respiration, and without, we were not. So if anybody has any idea why that might be, then shout. So survival strategies. So none of these are really novel carbon sources that we didn't know that M1 can use, but it shows that the biolog system and the model are in agreement. So there are 4 kind of groups here. There's the amino acid groups, which are broken down. So this is this group here into the TCA cycle. And creates energy. Then there's the TCA cycle intermediates, the fermentative products, which are normally byproducts of other bacteria. And then finally pyruvate, which is the metabolic hub, where everything is most things are broken down into before entering the electron transport chain. So what struck us as interesting with this is just that a lot of organic acid washes are being suggested as interventions to reduce Campylobacter, and they do reduce Campylobacter loads. However, what we're seeing is that with this strain, it's actually allowing it to respire. So this requires more investigation, and we think that maybe we should use this model to find acids which aren't which would never be used by Campylobacter to begin with. So in conclusion, M1cam was able to respire and survive on hydrogen, and some of these organic acids need more investigation in terms of food safety. And finally, I'd like to thank all of this group of people here, so Andrew and John especially, who allowed Polly and I to carry on with this project. Thank you so much.
Thanks, Emily. It was a really nice talk. We have a question in the chat from Aidan. So it says, I understand your explanation of why the conditions were different for the plus-minus hydrogen plates, but 5% versus 10% oxygen is quite a significant difference and going to influence the respiration rates and therefore your dye values. Did you do a control experiment with your plates at the different oxygen but without hydrogen in either? Thanks.
No, we actually didn't, but that would probably be a really good idea. The good thing about the system with the model is that the model can take into account these changes. But obviously I do understand that it's not therefore completely comparable.
Okay, Aidan is typing. We have another question from Dave Kelly. The electron transport chains in your model do not look like those that Campy actually has. This might affect the way you think organic acids are respired.
Okay, maybe Polly and I should have a chat with Dave then.
Yeah, you can actually continue with the chat in the Discord. Thanks a lot, Emily.
No, thank you.